did was, well, I named it the Who Are You? The Human Microbiome. I'm Vanessa, and that is Dr. J, my mentor. And what we did was we took 7 different samples, 2 from belly buttons, which we'll go into more, the tongue, the armpit, nose, throat, and ear. We're focusing more on the 2 belly button piercings though, because technically we have 12 in total. Because cooler. We did a pierced belly button and a non-pierced belly button. So right here this is the pierced belly button, and this is the non-pierced. And already from the sample that you can tell there's different bacteria that are growing within them. And
that's a slide.
What we first did was a gram stain. So right now we're just looking at the physical characteristics of them. So the gram stain is we could either do, it's kind of allows the bacteria to become colored, and from being colored we can tell the cell wall shape, which gives us whether or not it's a gram positive or gram negative. And we can also tell the shape and identify it from there. So for gram positive, it has a violet color. The reason why is because there's a thick layer of peptidoglycan. I'm not an English major, so please bear with me with these words. And from there, it traps, it kind of accepts the crystal violet, and it accepts an iodine solution. And then when alcohol is created, it closes the cell wall, dehydrating it and keeping the crystal violet color. While the gram-negative stain has a layer each of liposaccharides and lipoproteins, and a thin layer in between of the peptidoglycan. So when the crystal violet layer and the iodine make a complex together, the alcohol kind of gets rid of it, decolorizes it, and then a saffron solution colors it and makes it into a pink or red color. And then the pictures of it is going to be great. So let's do the easier one first. This is the non-pierced belly button. It has a cockside, which means that it's circled, and it's more of a red color. As the pierced belly button has cockside and rods. Rods are not really normal within a belly button, as you can see from there. So as we can already tell, like this is going to be a multi-bacterial solution, which is great. So then we do DNA extractions. It's a 45-minute process, so we're not going to get all into that. But this is basically the tools that we use, what's going on with it. And at the end, we get 2 microliters of DNA, which all end up in this.
Wear your glove,
So he's raising into that.
Good. All the procedure.
They're harmless, they won't kill you.
Right now.
Yeah, he told me that too. They're not harmful at all. These are just all the these are all the little things that are inside us, basically. There's a little less than 2 microliters though, because we had to use them for the following experiments. Which include DNA quantification, which tells us how much DNA is within the sample that we have here. For this, we had to use 2 samples that are provided for us, which gives us that. And then the belly button and the pierced belly button, as you can see, we already have a lot more pierced belly button, which is another, what is this, half an hour process, which I don't want to go into. And then we ran a gel sample. So we used a 16S RNA, which is a universal, because usually gel samples are to find a specific DNA sequence. So we just did a universal one so that we can just kind of have like a line going down. And we want to focus on the second and the third one. So the second one here is the belly button, the regular one, and then the third one is the pierced belly button. And what happens is that it's going down. So the DNA is negative, and here's a positive. Yes, negative, positive. And what happens here is this really big one here is a marker for 1,500, which is the molecular weight of that one part right there. And each one is like little gaps, the lowest one being the bottom marker where the most positive is, and the highest. And then we ran it to a lab, and we got these beautiful sequences. All the A, T, G, the sequences of both of them. The ends, I just want to add in there, the ends are the lab's way of saying, I don't know what this is, so I'm not going to guess. And then from there, we can go into a beautiful thing called the internet. And these 2 websites give you you can copy and paste the previous. So you can copy the belly button, let's say, go into one of these, and they give you what it is. They tell you what it is from the thing. So from the belly button, we know now that it is, I'm going to these names again. Acetylbastard Johnson strand B2. Let's go with that. And the identity here is 98%, which means that we basically know where it is. And the coverage is how much it's going. And then for the same thing, for the Pierce belly button is another bacteria, which is the Staphylococcus aureus strand Purgos. Great, with a 97% identity. Yes. And now what we're doing is we're going into the what's resilient to. So for this, let's see, this one's the non-pierced. This is right here the cytomycin, and this is the penicillin, and then right over here, this is cyclomycin, and this is the penicillin. And we also use peppermint oil and tea tree oil. We found out that Dr. J told me that there was like an article saying that a lot of like natural oils are actually really good like killers of certain bacteria, which is amazing and really cool. But I only focused on these two right now, just for the sake of this project. But as you can see here, the cytomycin here is really small. It's not really resilient, while as this one has a huge surface area, well compared to this one at least. So we can tell here that the non-pierced belly button is less resilient to the cyclomycin than the pierced belly button is. And that is so far where we have gotten to our, that's as far as we've gotten through everything. We still have a few more that we want to go to. And then carrying on to summer 2, we wanted to go more into the E. coli bacteria, not like the bad one, the one that lives in the large intestine of all mammals, that one, the safe one, right? Okay, good. I'm like scared that he's gonna secretly pull like a dangerous one on me, just putting this out there. And then I tried using Blogspot, but that hurt me, so I used the Wix site. And then if you ever want to contact us, we're basically always here. And yes, thank you.